Hi Imanema, my name is Sharon Letia, but you can call me Tia. I am currently doing my PhD in MPM, which is led by Professor Florian Grundler, and a big part of our work focuses on molecular interactions between plant parasitic nematodes, mostly Heterodera shaktii and Melodegain incognita, so that we can understand what happens when the nematode comes into contact with the plants. So Professor Wim asked me to make a short video on the model system that we use, which is the Heterodera shaktii and the Arabidopsis thaliana system primarily, though we eventually also use Melodegain incognita based on the demands of the experiment. So I'm going to divide this video into two, and the first part is going to be dealing with why do we choose to use the model plant of Arabidopsis thaliana as opposed to maybe rice which some labs already doing. So the first thing that I'll say is that Arabidopsis thaliana is actually a small plant, right? It means it takes less space when you grow it. You don't need a whole greenhouse to put Arabidopsis thaliana, which actually saves on space and eventually money, because if you have to construct a whole greenhouse and whatnot, so the plant is very small and very easy to handle. So I have, for instance, here Arabidopsis thaliana seeds. There could easily be 1,000 seeds here. The seeds are so fine. Maybe you can see from the back, the seeds are so fine that almost 1,000 seeds can fit in this one petri dish. So it means it makes it easier to work with, and also the plant itself is quite small. So we were able to put 2 plants in this petri dish, but it's because we choose to put 2 plants depending on the demands of your experiment. Sometimes you can put more, but this is the entire plant. This is even ready to flower right now. So we can see that the plant is very small and very easy to manage. Secondly, Arabidopsis thaliana has a very short life cycle. So on average, under optimal conditions, we're talking 6 weeks. And this becomes very important because if you want to study transgenerational things like maybe yield or like sizes of the seed pinnacles and whatnot, but I think this is really important for us when it comes to like making mutants, because oftentimes you need at least stable lines of up to 3 generations or even 6 sometimes. So it means if you're able to plant your if you're able to transform your plant and then allow it to grow for just 6 weeks, then it means the first generation is ready. Then you can harvest this first generation and then seed it again to start your second generation. So it means in juxtaposition to other plants like maybe banana or even maize, then you see that the lifespan is so short that it allows you to work faster. So for example, you could be able to get a stable mutant in under 1 year compared to another annual plant or a perennial plant that may take longer. So another thing that I can think about is that Arabidopsis has a relatively small genome, which has also been thoroughly well characterized. You can find a lot of resources on the Arabidopsis plant based whatever you're looking for. If you want to find transcriptomic data on its interaction with nematodes, on its interaction with light, on its interaction with salt, and its interaction with water and drought, chances are it's out there. So if you just have good mining techniques, you can be able to actually generate a lot of data even without doing some of these experiments yourself. So on an experimental basis also, another advantage I can think about is that Arabidopsis actually is a host for a decent number of pathogens. So like I could think about bacteria, I could think about viruses, I could think about fungi, I could think about plant parasitic nematodes and different genera of these that are actually able to parasitize Arabidopsis thaliana. So this is particularly important because you have one plant that is able to be parasitized by different pathogen, and this is important because sometimes you do experiments and you find out of maybe a mechanism, right? And you want to understand, is this mechanism species specific, or this mechanism is actually seen in root knot nematodes and it's also seen in cyst nematodes. Okay, if it's seen in cyst nematodes and root knot nematodes, is it also seen in bacteria or is it seen in fungi? So imagine you have this one plant that you can just keep dumping all these other pathogens on, and you're able to get results without necessarily having to optimize another system which will accommodate your new pathogen or your side pathogen that you want to investigate. So it's really important that this one plant can be parasitized by a wide range of pathogens, right? Another thing that I can think about is actually the fact that Arabidopsis can grow very easily on different types of media. I'll give our lab for example. We grow Arabidopsis both on a media we call knop media and another media we call MS media. So the only reason we change this media is to fit the preference of the nematode. We do not know why, but like for example, Heterodera shaktii just prefers knop media, and Melodega incognita somehow just prefers MS media. So if we have this one plant that can grow on these different types of media, then it becomes easier. It's like one size fits all, right? So that's another advantage of Arabidopsis, and maybe this is more particular to nematode work, but I would think the anatomy maybe, or the root structure of the plant itself is actually quite important. I'll show you for instance that this is the root of Arabidopsis thaliana, and you can see that it's quite transparent actually, and this becomes very important when you're trying to do microscopy or cellular work, because then again, I'll give you an example of one experiment we do in our lab. We have an experiment where we actually investigate very early interactions, and one of that is initial syncytial cell selection. And the only way you can tell that this nematode has selected its initial cell is when the stylet stops pumping, because what happens is that when the nematode enters into the cell, then it starts to pump effectors, which you can actually really see that the stylet is moving like this. It's pumping, it's pumping, it's pumping. So once it stops, it means that the hard work is done, and now it's just waiting for the effectors to maybe change the genetics of the plant, or like to stop some things, so that then the actual syncytia starts to grow, right? And the transparency of this root, for example, makes it very, very important for us to see through the petri dish, through the media, and through the root to actually see the stylet of the nematode. So this could only be limited to nematode research, but it's really, really important. And it's also important for things like microinjections. If you want to do experiments that involve really fine experiments like that, it's important that you see where you're going, and the transparent roots make all the difference. So I think those are some of the advantages of using Arabidopsis thaliana as a model plant, especially for interactions between nematodes and plants. So the second part of this video is about sterile work. So as you may already have seen, that both the seeds and the sample plant, they are quite sterile. I can tell you certainly that this is a very sterile sample. The petri dish inside was autoclaved. The seeds were sterilized, and the petri dish was sterile. and the work was done under a clean bench, right? And you can also see that once the plants have grown, you don't see any form of infection, and yet these growth mediums are quite rich. So it means if there was presence of any external bacterial cell, fungal cell, they would easily proliferate in here because these media are nutrient rich, right? But you can see, for example, this is a very, very sterile petri dish, right? So the question then is, why do we work under sterile conditions? So I could start by saying that the plant in itself is not so highly evolved that it will have very unique immune responses that are for bacteria alone, for nematode alone, and for fungi alone, for example, right? Sometimes we see that the mechanism of perceiving these pathogens may actually be different. Maybe we know that flagellin is only identified by FLS2, and then maybe ascaricides is perceived by NILR1, according to our recent research. But then we have also seen that sometimes even some of these pathogenic cognition receptors are promiscuous. It means they're able to perceive more than just one pathogen. So the point here is we do not want to record false positives. So the point is, I will use human bodies, right? If you have malaria, for example, for the people from tropical countries, chances are you'll have a fever, right? But if you have a bacterial infection, you'll still have a fever. You'll have a viral infection and you'll still have a fever, right? So it means these are very different disease-causing pathogens attacking the body, but the response of the body is the same, right? So except you do diagnostics work, sometimes you may not be able to tell if truly this fever is from the malaria or from the typhoid or from the viral infection, right? So in order to have correct results, then we have to work with azenic cultures. So in our context, azenic is the fact that we only have one pathogen inside here, which will then be the plant parasitic nematode, right? Not even two parasitic nematodes, but just one. Per time, we're always working with either only Heterodera shaktii or only Melodegai incognita. So this is why it's very, very important for us to work under sterile conditions. We cannot have bacterial infections because, as it's already known in literature, that upon bacterial perception, there's a release of rust bursts. This is the same thing that we're seeing in plant parasitic nematodes. We also see that when bacteria is perceived, there's also hypersensitive reaction, which is usually characterized by callus deposition. This is the same thing that we see in nematodes. So you don't want to write a paper and say that, oh, upon perception of nematodes, we are seeing that there's an increased rust burst, and yet this rust burst in part may be due to the bacterial infection. So that's why it becomes very important for us to work in sterile conditions. I can just give you another final example before we wrap up. Think about the sky at night, right? The sky at night has stars and it also has the moon, right? So it means if you want to gauge the amount of light produced by the stars, if you have the moon in the sky, you're not going to have correct results. You're going to probably have an amount of light that is partly star, partly moon. So it means you're recording a lot of false positives inside there. So what you do in that situation is that if you want the light from the moon alone, you're going to have to remove the stars from the sky, and only then will you be sure and confident that the light you are measuring is solely from the moon and not from the stars. So in a nutshell, downstream responses of bacteria, nematode, viruses, and fungi are often quite similar. So it becomes important that you take out every other pathogen and only focus on your pathogen of interest. And that's why it becomes very, very important for us to work under sterile conditions so that we are not registering any false positives for parameters that we are looking for while we try and understand molecular interactions between plant parasitic nematodes and plants using our pathosystem of Heterodera Shaktii and Arabidopsis thaliana. So I'll be happy to answer any other questions that you have, and until next time, it's been a pleasure. Bye.